Okay, good morning students. In previous class, under the chapter Biotechnology, Principles and Processes, we will discuss regarding the diagrammatic representation of recombinant DNA technology and afterwards separation and isolation of DNA fragments. Separation and isolation of DNA fragments, we used to do the separation and isolation of DNA fragments with the help of the biotechnology in the sense technique which is used named as a gel electrophoresis, and afterwards we have discussed regarding the steps involved in a gel electrophoresis, stain used for the visualizing the fragments of DNA. After that we have discussed regarding the definition of elution, that is the last step. Okay, these are all the things we have discussed in a last class. In today's class, we are going to start with the cloning vectors. Okay, when we were discussed regarding the modifications in the sense processes in a biotechnology, that time we had discussed like origin of replication, cloning, plasmid, restricted endonuclease, DNA ligase and all. These all are the what tools which are used in a biotechnology. In these tools, I told you that the plasmid, it is act as a vector, it is going to carry the alien DNA into the host organism. So, regarding that vector, today we are going to discuss what are the features of vector and based on which features we are going to select the vector for transforming the alien DNA into the what we are going to call that as a host cell. So today we are going to start with the cloning vectors. Cloning vectors are nothing but you all means I think you know about the plasmid as well as bacteriophage. These are the act as a what we are going to call that as a cloning vectors. And these plasmids and bacteriophage both are having their own means have the replicating capacity. I told you that the plasmid it is an extra chromosomal DNA and it has its own replication, means replication capacity. So, the cloning vectors are the plasmids and bacteriophage, they are having the self-replicating capacity, and these bacteriophage, the number of these high number of per cell, in the sense bacteriophage are present in high numbers in a bacterial cell. As compared to the bacteriophage, the plasmids are few, in the sense 1 to 2 copies in a bacteria, or else in some time they are 15 to 100 copies per cell. 15 to 100 copies per cell. So, these plasmids are found in bacteria and bacteriophage also, those are found in a bacterial cell. So, when we are going to join the alien DNA to these plasmids or a bacteriophage and introduce into the host bacterial cell, that time what is going to happen? The plasmid is going to replicate, bacteriophage is going to replicate. That time your alien DNA, whatever you are having the interstate DNA base is joined to the plasmid and bacteriophage. That is also can replicate, and you can increase the number of that alien DNA bases. How the number is going to increase plasmid and bacteriophage per cell. Okay. Likewise, we are going to increase the interstate gene copies. So that is what the cloning vectors are used as a plasmid and bacteriophage. For this, you are going to join the foreign DNA. And most in biotechnology, we used to use the plasmid as a vector, cloning vector. Understood? So what is given there? Their numbers can grow even higher, in the sense, for example, if you are able to multiply its number equally to the copies of the plasmids and bacteriophage, in the sense if we are going to join the alien DNA to the plasmid or bacteriophage, once you are going to able to join the alien DNA to the bacteriophage or the alien DNA to the plasmid, when they start going into the bacterial cell, their number, how their number is going to grow, likewise your alien DNA also going to replicate and increase its what we are going to call that as a quantity. That is what the vectors, the cloning vectors are nothing but the plasmid and bacteriophage. Now, the next point we are going to study, that is the features that are required to facilitate cloning into the vector. that is required, the features that are required for the facilitating the cloning into the vector. Now here, what are the features which are required for the facilitating the cloning into a vector? Now here we are going to study the 4 different features of a cloning vector. That is very important. The 4 different features we are going to study here regarding the cloning vector. First one is origin of replication. Second one is selectable markers. Third one is cloning site. And fourth one is vectors for a cloning genes in a plants and animals. Okay. So cloning vectors are nothing but plasmid. What is my plasmid? Plasmid, it is a extra chromosomal DNA and it has a self-replicating capacity. In the sense, in today's class we are going to discussing about the cloning vectors. Cloning vectors are nothing but a plasmids and bacteriophage. Bacteriophage, it is different from the bacteria. And these two, what plasmids and bacteriophage have replicating capacity when they are there in a bacterial cell. Now, how we are going to introduce the alien DNA or recombinant DNA? We are going to choose the alien DNA in the sense origin DNA that we are going to join with the bacteriophage or else plasmid. Okay. Piece of alien DNA we are going to join to the bacteriophage or a plasmid. And once this plasmid or a bacteriophage with the alien DNA we are going to introduce in the bacterial cell, they are going to start replicating. And with them, the alien DNA is also going to replicate and increase the quantity of alien DNA. And I told you here, plasmids are present in a bacterial cell, normal bacterial cell, 1 to 2 copies or 50 to 100 copies. If there are 1 to 2 copies, then you are going to join the alien DNA to that, it starts replicating and it increases its number. And with increasing its number, the quantity of alien DNA is going to increase, and further we are going to extract from that bacterial cell. Right? Now then, we are going to study about the features that are required for the facilitating cloning into the vector. Cloning into the vector in the sense what? Either now alien DNA vector na hang join hang ಮಾಡಬೇಕು and the join ಮಾಡಿದ cloning in the sense similar copies you are going to get. Understood? ನಮಗೆ ಯಾವುದು origin DNA ಬೇಕಾಗಿತ್ತು ಆ ತರ copies ಮಾಡ್ಕೊಂಡು ಹೋಗೋದು. Multiply copies ಮಾಡ್ಕೊಂಡು ಹೋಗ್ತೀವಿ. So ಆ cloning ಮಾಡಬೇಕಾದ್ರೆ vector ದ ಏನೇನು features ಬೇಕು? What are the features which are required for facilitating the cloning into the vector? In the sense plasmid ಏನೇನು features ಇರಬೇಕು that can facilitate cloning into the vector. Right? So that is, first one is origin of a replication, second one is selectable markers, third one is cloning site, fourth one is vector for cloning genes in a plants and animals. So sometimes the 5 marks question is going to ask based upon this, write the features that are required for the facilitating cloning into the vector. If you are going to write only the site headings, you are going to get 2 marks. Half up. What is that? Origin of replication, selectable markers, cloning site, vector for the clone genes in plants and animals. Now, here the origin of replication in the sense you all are aware about the DNA is double stranded and if it is going to make its own duplicate copies, that is what we are going to call that as a semi-replication. Okay? Alena replication it start unwinding at certain point. It starts unwinding at a certain point, and that we are going to call that as a origin of replication. Understood? Alien active pass understanding value, then the duplicate copies are going to form of a DNA. That is what origin of replication. We are going to call that one as a shortly ORI. This is the sequence from where the replication starts. The replication starts from a particular sequence. Which gene we are interested or which protein is going to form, based upon that, the sequence there, the replication is going to start. We are going to call that as a origin of replication. And this sequence is also responsible for controlling the copying number. Controlling the copying number in the sense what? Yes, the copy weight number. For example, 1000 500 whatever it may be. Based upon that, this replication is going to control how much copies we require. And next one is to the link the DNA. Now I have the link material. So this is about what? Origin of replication. Origin of replication. In the sense, for example, I am going to draw the plasmid here like this. This part one point origin of replication. From that, the replication is to start. Okay, afterwards I am going to draw in detail regarding the plasmid diagram. Next point, after the origin of replication, we are going to study selectable markers. We used to use the selectable markers for the cloning into a vector. In addition to ori, the vectors required a selectable markers. The vectors required a selectable markers. And what these selectable markers are used to do? In the sense, these selectable markers helpful to identifying and eliminating the non-transformant. For example, transformant ಅಂತಂದ್ರೆ ಏನು? ನಮಗೆ ಯಾವುದು ಜೀನ್ ಮುಂದೆ ನಮಗೆ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡೋದೈತಿ ಮತ್ತು ಯಾವ ಜೀನ್ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡೋದಿಲ್ಲ ಅದಕ್ಕೆ ಏನಂತ ಕರಿತೀವಪ್ಪ ನಾವು ಅಂತಂದಾಗ ಒನ್ ಈಸ್ ಟ್ರಾನ್ಸ್ಫಾರ್ಮರ್, another one is non-transformant. If you are not going to use selectable markers, ನಾವು selectable markers ಯೂಸ್ ಮಾಡಬೇಕು. ಆಗೇನಾಗಪ್ಪ ಅಂತಂದಾಗ ಟ್ರಾನ್ಸ್ಫಾರ್ಮರ್ ಜೊತೆ ನಾನ್ ಟ್ರಾನ್ಸ್ಫಾರ್ಮರ್ ಕೂಡ ಏನಾಗ್ತವು ನೆಕ್ಸ್ಟ್ ಏನಪ್ಪ ಅಂತಂದಾಗ ಕ್ಲೋನಿಂಗ್ ನ ಹೋಗ್ತವು. So these what we are going to call as a selectable markers are useful or helpful to identifying and eliminating the non-transformant. They used to identify and eliminate the non-transformant and selectively permit the growth of the transformant. They used to permit the growth of the transformant. Andre for example ಈಗ ನೋಡ್ರಿ ಈಗ ಒಂದು A ಒಂದು B ಐತೆ ಅಂತ ಇಟ್ಕೊಂಡು A is non-transformant, B is transformant. If you are going to use these selectable markers, they are going to identify the A and they are going to eliminate that A. Understood? And permit B to transfer. Permit B to transfer. That is the function of selectable markers. Now here transformation. What is meant by transformation? It is a process through which a piece of alien DNA introduced into the host bacteria. Piece of alien DNA introduced into the what we are going to call that as a host bacteria. If suppose now bacterial cell ಅಂತಂದಾಗ bacterial cell ಅಂತಂದಾಗ suppose is a bacterial cell ಇದ್ರೊಳಗೆ ನಾವು ಏನು ಮಾಡಬೇಕು alien DNA introduce ಮಾಡಬೇಕಾದ್ರೆ direct introduce ಮಾಡಂಗಿಲ್ಲ. First we used to join this alien DNA to the what vectors. Vector in the sense plasmid. Then you are going to introduce into the bacteria. Right? Now here the selectable markers example in the genes encoding to the antibiotic resistance. Antibiotic resistance ಅಂದ್ರೆ ಏನೀಗ antibiotics ಕೊಡೋಣ ಅವರ ಬಗ್ಗೆ ಫೈಟ್ ಮಾಡುವಂತ ಒಂದು ಏನಂತಾಗ genes. What are those ಅಂತಂದಾಗ one is ampicillin, chlorophenol, the next one is tetramycin or kanamycin. These are what the genes which are resistant to the what we are going to call it as a antibiotics. But these all the genes are not found in a normal E. coli bacteria. E. coli bacteria ಇರಂಗಿಲ್ಲ. ತಿಳಿತಾ? ಈಗ ನಮಗೆ ಏನು ಮಾಡೋದೈತಿ ಆಂಟಿಬಯೋಟಿಕ್ಸ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಜೀನ್ ನ ತಯಾರು ಮಾಡಬೇಕಾದ್ರೆ ನೀವು ಇದನ್ನು ಇವೇನು ಆಂಟಿಮೈಸಿಲಿನ್ ಕ್ಲೋರೋಮೈಸಿನ್ ಇದಲ್ಲ these things you have to join to the plasmid. ಆ ಪ್ಲಾಸ್ಮಿಡ್ ನಾವು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಸೆಲ್ ದಾಗ ಹಾಕಿದಾಗ then they are going to replicate and there we are going to find these all the genes. ಅರ್ಥ ಆಯ್ತಾ? ನಾರ್ಮಲ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಇರಂಗಿಲ್ಲವ, ನಾರ್ಮಲ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ ನಾವು ಹಾಕಬೇಕಾಗ್ತೈತಿ ಎಲ್ಲಿನ ಹಾಕೋಣ ವಿತ್ ದಿ ಹೆಲ್ಪ್ ಆಫ್ ಪ್ಲಾಸ್ಮಿಡ್. ಆನಂತರ ಈ ಪ್ಲಾಸ್ಮಿಡ್ ಹೆಂಗೆ ಗ್ರೋತ್ ಆಗ್ತೈತಿ ಅದೇ ರೀತಿ ಇವೇನು ನಮ್ಮ ಜೀನ್ಸ್ ಅಲ್ಲ ದ ಜೀನ್ಸ್ ಕ್ವಾಂಟಿಟಿ ಆಲ್ಸೋ ಗೋಯಿಂಗ್ ಟು ಗ್ರೋ. ಅವು ನಾವು ನೆಕ್ಸ್ಟ್ ಏನಪ್ಪ ಅಂತಂದಾಗ ಫಾರ್ ಮೀನ್ಸ್ ಫಾರ್ಮೇಷನ್ ಆಫ್ ರೀಕಾಂಬಿನೆಂಟ್ DNA ಮಾಡೋಕೆ ನಾವು ಯೂಸ್ ಮಾಡೋದು. ದಿಸ್ ಇಸ್ ಅಬೌಟ್ ವಾಟ್ the selectable markers. Now next used to come that is the cloning site. Cloning site in the sense what? ಈ cloning site ನಾಗ for example ಇಲ್ಲಿ ಒಂದು ವೆಕ್ಟರ್ ನಾಗ ಒಂದೇ ಸೈಟ್ ಅಂತ ಇಟ್ಕೊಂಡು ಕ್ಲೋನಿಂಗ್ ಎದಕ್ಕೆ ಮಾಡಾಯ್ತದ ಆಂಟಿಮೈಸಿಲಿನ್. that time what is going to happen ಅಂತಂದಾಗ it is easy to clone and produce the more number. If suppose there are more than 2 or many cloning sites are there, ಬಹಳಷ್ಟು cloning site ಇತ್ತಂದ್ರೆ ಏನಾಗ್ತೈತಿ there is bit confusion which is going to what select and which is not going to select. Understood? In the sense vector ಹೆಂಗಿರಬೇಕಪ್ಪ ಅದನ್ನ vector should be consist minimum cloning site. Minimum cloning site ಅಂದ್ರೆ ಏನ ಒಂದೇ ಇದ್ರೆ it is very good. Okay, minimum 2 or 3 more than means more number are present in cloning means in a vector regarding the cloning site that is not good for the formation of recombinant DNA. Understood? ಹೆಚ್ಚಾತಂದ್ರೆ ಏನಾಗ್ತೈತೆ ಒಂದು ಸ್ವಲ್ಪ ಏನಪ್ಪಾ ಅಂತಂದಾಗ if in the sense preferable ಏನಿರ್ತೈತಿ few cloning site ಇರಬೇಕು. ಒಂದೇ ಇತ್ತಂದ್ರೆ it is well and good. Understood? Regarding the cloning site we are going to discuss in detail in next class. And last is to study the vectors for a cloning gene in a plants and animals. Plants ಮತ್ತು animals ದಾಗ ಯಾವ vector ಯೂಸ್ ಮಾಡ್ತಾರೋ that can be useful for us to making the cloning what we are going to call it as a to forming the cloning. This is about what? The origin of replication, selectable markers, cloning site, and vector for the cloning genes in plants and animals. Understood? Yes, to bracket formula. This is asked for the 5 marks. And sometimes 2 marks question is going to ask. 2 marks question is going to ask. That is what? Name the genes which are encoding for the antibiotic resistance. 2 marks ಗೆ ಇರುತ್ತಂದ್ರೆ ನಿಮಗೆ 4 ಬರೆಯಕ್ಕೆ ತಿಳಿತಾ? 1 marks ಗೆ ಇರುತ್ತಂದ್ರೆ 1 or 2 used to write. Understood? This is regarding what? The cloning vector. Name that is a plasmid and bacteriophage. They are having a self-replicating capacity, and these are found in a normal bacteriophage. The plasmids are less number as compared to the bacteriophage. Now the plasmids are used as a vector. Understood? Then we are going to study about the features that are required to facilitate cloning into the vector. Those are origin of replication. That is what we are going to call it the ORI. Second one is the selectable markers. Selectable markers in the sense genes including means encoding to a antibiotic resistance. What are those? Ampicillin, chlorophenolone, then tetramycin and kanamycin. These are very important. You are going to remember these things. Antimycin, chloro, ampicillin, tetramycin and kanamycin. This is why 2 marks question bahಳ ಸತಿ ಕೇಳಾರೆ. ಆನಂತರ ಇವು ಎಲ್ಲಿ ಇರಂಗಿಲ್ಲ ಅಂದ್ರೆ ನಾರ್ಮಲ್ ಈ ಕೊಲೈ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಇರಂಗಿಲ್ಲ. Understood? Now the plasmid flow introduce ಮಾಡಬೇಕು. Then in next class we are going to study about the cloning site and it is for the cloning genes in a plants and animals. And the homework for you people, you are going to draw a neat label diagram of plasmid. Plasmid ಏನು ಮಾಡ್ತೀವಿ ನಾವು ಡಯಾಗ್ರಾಮ್ ತೆಗೆದು ಆ ಡಯಾಗ್ರಾಮ್ ನ ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸ್ ನಲ್ಲಿ ಮತ್ತೆ ಡ್ರಾ ಮಾಡಿದಾಗ ನೋಡಬಹುದು. ಓಕೆ ರೈಟ್, ಐ ಕಂಟಿನ್ಯೂ ನೆಕ್ಸ್ಟ್.